In this video, we're going to look at the regulation of the citric acid cycle. So before we get started, it's a good idea to take just a minute, maybe pause the video, and review the different steps of the citric acid cycle, including the precursor step or the entry into the citric acid cycle with acetyl-CoA. So let's take a look at how the citric acid cycle is regulated. There are three major points of regulation in the citric acid cycle. The first and major regulation point is control of flux into the citric acid cycle. So basically, the control of the pyruvate dehydrogenase complex. The second important step is isocitrate dehydrogenase. And the third is alpha ketoglutarate dehydrogenase complex. So these are the three major points where our citric acid cycle is regulated. Now, if you look at the figure here, there's a lot of different things going on. So let's take just a minute to try to make some sense of this before we try to get into this in any detail. So there's really three major things that control the citric acid cycle. First, our enzymes are generally regulated by ATP and ADP. Not all of them, but some of them. So if we have, let's say we have a high concentration of ADP, what does that tell us? It tells the cell that most of the ATP has been hydrolyzed and we need to make more ATP. So what do we want the citric acid cycle to do? Well, we want it to be very active. So ADP tends to activate some of our enzymes, such as isocitrate dehydrogenase and the pyruvate dehydrogenase complex. And of course, on the other hand, if we have high ATP concentration in the cell, that tells the cell that we don't need to keep making ATP. We've got enough energy. So high ATP concentrations tend to inhibit enzymes like pyruvate dehydrogenase complex and isocitrate dehydrogenase. Now, there's another important control molecule for our citric acid cycle, which of course would be NAD plus and NADH, because that's one of the major roles of the citric acid cycle is to produce these reduced electron carriers. So the redox state is the citric acid cycle is very tuned into the redox state of the cell. If we have high NAD plus concentrations, that means we don't have a lot of NADH. So we need to make more. So NAD plus is an indicator that we need to make more. And NADH, when we have high NADH concentrations, that tells the cell that we've got plenty of energy. We've got plenty of NADH. We don't need to keep making it. So NADH tends to inhibit our major regulatory enzymes, as you can see here. And thirdly, inhibition of relevant enzymes by acetyl-CoA and succinyl-CoA. So again, if we have a high acetyl-CoA concentration within the cell, that tells us that we've got plenty of energy, we've got plenty of intermediates, so that tends to inhibit the citric acid cycle at the pyruvate dehydrogenase complex. And if we have high succinyl-CoA, which is again a very high energy compound, that would tend to inhibit our alpha ketoglutarate dehydrogenase complex and also our pyruvate dehydrogenase complex. So when you see these inhibitors and activators, think of what do they indicate that the cell has a lot of energy? If so, it tends to inhibit our citric acid cycle. Or do they tend to indicate that we have low energy state or low redox state, and therefore we want to activate the citric acid cycle? Now, for our purposes, we really want to just focus on the major regulatory point, which would be our pyruvate dehydrogenase complex. So that's what we're going to talk about in this video lecture, regulation of the pyruvate dehydrogenase complex. And its regulation is actually quite simple. For one, it is activated by its substrates. So if we have, remember the substrates, pyruvate, NAD plus, and coenzyme A, and the products, NADH, acetyl-CoA, and carbon dioxide. If we have a high concentration of NAD plus, it's going to activate our complex, and it's going to activate it at the E3 portion, because that's where NAD plus binds. And if we have a high coenzyme A concentration, it's going to activate it because, again, it's a substrate, so it's going to activate it at the E2 portion, because that's where that molecule binds. And it tends to be inhibited by high concentrations of the products. So if there's a high concentration of NADH, like we said, it tells the cell we've got plenty of energy, plenty of these reduced electron carriers, so it will tend to inhibit our pyruvate dehydrogenase complex at the E3 portion. And if we have high acetyl-CoA concentrations, it will tend to inhibit our enzyme by binding at the E2 portion. So our pyruvate dehydrogenase is complex activated when substrate concentrations are high and inhibited when product concentrations are high. Now there's one other form of regulation of our pyruvate dehydrogenase complex, and that involves phosphorylation. Phosphorylation is a very common means of activating and inactivating enzymes in different metabolic or signal transduction pathways. And that's true of our pyruvate dehydrogenase complex. The E1 portion of our complex can be phosphorylated by a particular enzyme called pyruvate dehydrogenase kinase. Remember, kinases add phosphate groups. So when pyruvate dehydrogenase kinase phosphorylates our pyruvate dehydrogenase complex, it will inactivate it. So phosphorylation inactivates our complex. Now, here in our figure, you can see the E1 portion represented here. Here is a serine residue, and we have a phosphate added onto that. So here, this would represent our phosphorylated E1 portion, which would inactivate our pyruvate dehydrogenase complex. And again, the PDH kinase is what adds that phosphate. Now, what activates the pyruvate dehydrogenase kinase? Well, it tends to be activated when we have high ATP, NADH, and acetyl-CoA. Well, what do those, again, all of these indicate that we have high energy, high redox state, high intermediate, so we don't need to keep the citric acid cycle going. So when these intermediates or molecules are high in concentration, they will tend to activate, as you can see here, our kinase, and the kinase will phosphorylate and inactivate the pyruvate dehydrogenase complex. So again, be sure you keep that straight. It can be a little tricky. So when the kinase is activated by a high energy state in the cell, it will turn off the pyruvate dehydrogenase complex and therefore shut off the citric acid cycle. Now, we don't want it shut off permanently, so there must be a molecule that, or an enzyme that will remove that phosphate. And of course, there are phosphatases which remove phosphate groups. This would be, of course, the pyruvate dehydrogenase phosphatase, which will remove that phosphate, and you can see that here. Here's our E1 complex. This is a serine side chain, and the phosphate's been removed. So our phosphatase will remove that phosphate, thereby reactivating the complex and thereby reactivating the citric acid cycle. And the phosphatase tends to be activated by high concentrations of ions and insulin, which of course gives this regulation a link to outside the cell. So everything's not just dependent on intracellular concentrations. Control of the citric acid cycle can be regulated by extracellular signals such as insulin. So our pyruvate dehydrogenase kinase, when it's activated by these high energy molecules, or let's call them molecules that represent a high energy state, our kinase is activated, it phosphorylates, it turns off the pyruvate dehydrogenase complex, shutting off or slowing down the citric acid cycle. When we have a stimulation by insulin, it will tend to activate the phosphatase, which will turn on the citric acid cycle, thereby increasing flux through the citric acid cycle. So that's the regulation of our pyruvate dehydrogenase complex. Take some time, be sure you're familiar with those steps.